Welcome back to our video on Gametogenesis. In the first video we saw spermatogenesis, but in this one we'll be looking at oogenesis. Oogenesis involves the production of the ovum or egg of the female from primordial germ cells, just as we saw in the case of the sperm cells. Now, if we have the primordial germ cell like this, let me write that as GC. So if you remember, in the case of the male, we said the primordial germ cells are on the inner lining of the testis. In the case of the females, you find them on the inner lining of the ovaries. These primordial germ cells are usually there even before birth, before a female child begins to grow at all. The child already has the primordial germ cell, and they even undergo some development even before and during childhood. So that for the primordial germ cell, by mitosis, just as is occurred in spermatogenesis, primordial germ cells become what we call oogonia, and then by growth, the oogonia become primary oocytes. Primary oocytes. Now, this growth we're talking about here is just as it occurred with the spermatogonia in spermatogenesis. But we say this growth is much more significant, like there's a very large amount of growth, so that the egg cell is many times bigger than the sperm cell. You know, before we said that the sperm cell is about 2.5 micrometers long. Now 0.1 millimeters, which is many times the other value, is what we have as an estimated value for the diameter of an egg cell. So the egg cell is by far a larger cell than the sperm cell, and that's because of the large amount of growth taking place here. So that for the primary oocytes, they will eventually undergo what we call meiosis 1. So by meiosis 1, they are going to become secondary oocytes. In terms of chromosome number, this is still diploid, this is diploid growth, so it will still be diploid, but meiosis 1 gives rise to haploid daughter cells. So while the primary oocyte is diploid, the secondary oocyte is haploid, and that secondary oocyte is formed from the primary during meiosis 1. Now, if you know about meiosis 1, or if you know about meiosis generally, you realize that meiosis is divided into, okay, let's do this now. For mitosis, you know, it's just in one stage. It's unlike in meiosis where we have meiosis 1, meiosis 2. So that for mitosis, we say that there are PMAT stages. What are PMAT? Prophase, metaphase, anaphase, telophase, and mitosis is complete. But for meiosis, what we have is prophase, metaphase, anaphase, telophase, followed by prophase, metaphase, anaphase, telophase. So that this PMAT is called meiosis 1, and this PMAT is called meiosis 2. So that the prophase here is called prophase 1, but the prophase there is called prophase 2, and so forth. So that during this meiosis 1 we're talking about here, there's prophase 1, metaphase 1, anaphase 1, and telophase 1. The meiosis 1 is not completed in an individual, in a female child, until that child reaches puberty. So see this, at the point of birth and during childhood, what a female child has in her ovaries is not primordial germ cells, neither is it oogonia. Instead, it is primary oocytes that have started meiosis 1 but have not gone beyond prophase 1. So we say that the primary oocytes have started undergoing meiosis, but the meiosis has been arrested at prophase 1. That's what we have in a young girl of say 4 years old, 5 years old, that has not reached puberty. She's carrying primary oocytes already, and those primary oocytes have started meiosis 1, but that meiosis has been arrested at prophase 1. So that by puberty, every month, one of these primary oocytes gets selected for development. So that is what we refer to later, or we'll talk about later as the female cycle and so on. So that when that primary oocyte is selected within a particular month like that, it will now undergo metaphase 1, anaphase 1, telophase 1. And once it undergoes those, we say that meiosis 1 is complete. And then we have a secondary oocyte. Now that secondary oocyte will undergo what we call meiosis 2. Again, meiosis 2 has its own prophase, metaphase, anaphase, telophase. Now this meiosis 2 that this secondary oocyte is undergoing will not be completed. Instead, it will be arrested at metaphase 2. So it's as though what I'm trying to tell us now is, for a female that has reached puberty, what she has in her ovaries are primary oocytes arrested here at prophase 1. Then, every month, each of these primary oocytes will be, as in one of them rather, would be collected or selected and will continue its development to become secondary oocyte and even start meiosis 2, but only complete metaphase and then prophase and then get arrested at metaphase. So that it is at this stage that the egg is released. When we talk about the release of an egg, they say, oh, a woman has carried out ovulation. A woman has released an egg. That egg she is releasing is actually not a mature egg. Instead, what she is releasing is a secondary oocyte on its way to becoming a mature egg, but paused at metaphase 2. Because actually, this second meiotic division is supposed to convert this secondary oocyte into what we call an otid. Just as you have the spermatid, this is supposed to be an otid. So we are saying that somewhere here, there's a pause, prophase 1, and that pause will remain in the female until she reaches puberty. Then by puberty, once a particular egg is selected, once a particular primary oocyte is selected, that primary oocyte will continue all the way to secondary oocyte and then all the way to metaphase 2 and pause until fertilization. So when there's ovulation, what a female releases, like we said, is a secondary oocyte paused at metaphase 2. And that secondary oocyte is what will remain in the fallopian tube of that woman, waiting for a sperm to come and fertilize it. If a sperm does not come, that secondary oocyte dies as a secondary oocyte paused at metaphase 2. But in the event of a sperm coming to fertilize that secondary oocyte, then the secondary oocyte will quickly complete its development by undergoing anaphase 2, telophase 2, and then becoming the otid, which then matures into the ovum, and then this ovum gets fertilized. So fertilization is the point at which the rest of this process is completed. Otherwise, without fertilization, the secondary oocyte that is stuck at metaphase 2 dies that way. It just goes away that way. But if there's a sperm, then it can continue all the way to ovum. Now, let me mention here that meiosis 1, of course, produces a secondary oocyte, but that's not all. It also produces a small structure here that we refer to as a polar body. Now, this polar body is very tiny compared to the egg. Imagine something like this dividing into this and that. So the polar body is very tiny. It is insignificant. As a matter of fact, it is going to degenerate eventually. But its purpose is possibly to reduce the chromosome number, like from 46, 23 enter here and then 23 enter there. So we say the purpose of the polar body is to reduce the chromosome number. Then this meiosis 2, the secondary oocyte becomes an ootid, yes. But apart from the ootid, there is also another polar body. So this is also a polar body. And as such, we want to call this one second polar body, while we call that one first polar body. So in meiosis 2, the secondary oocyte divides to give an ootid and a second polar body. And it's also possible that during that same meiosis 2, the first polar body, which was formed along with the secondary oocyte that has just divided now, will also divide. So it's possible for this guy to divide during meiosis 2, because the two of them are the products of meiosis 1. So while this one is dividing into ootid and second polar body, this one too may divide to give two more polar bodies. So which means by the time we are getting an ootid, it's either we have two polar bodies or three polar bodies. And in that case, if we have three polar bodies, one of them will be the second polar body, then the other two will be the division products of the first polar body. In any case, the ootid matures to become an ovum. So this is just a representation of the stages, and the names sound familiar in that they resemble the names we heard when we looked at spermatogenesis. But I tell you, this is way more complex. It is way more complex in different ways. There are so many ways in which this one is more complex, especially in the area of hormonal regulation. So let me say a word on hormonal regulation of oogenesis. How is oogenesis regulated? Which hormones regulate this process? The hormones include, again, GnRH coming from the hypothalamus, and then we have FSH and LH, again coming from the anterior pituitary, and then we have estrogen, and we have progesterone. Estrogen and progesterone. So these are the main hormones that are concerned with this process. And what is their role? What do they do? I'll begin with FSH. FSH produced in the anterior pituitary. Of course, we said that its production will be under the influence of GnRH. So if GnRH is released from the hypothalamus, it will stimulate the anterior pituitary to release FSH. When this FSH is produced, it will go to the ovary, and when it reaches the ovary, that's FSH now, it will stimulate the selection of one of these primary oocytes paused at prophase 1 or arrested at prophase 1, and then it will stimulate cells to gather around it. So cells begin to gather around. Let me shade that. So what you see shaded there is the primary oocyte. So cells will surround it like this, and as those cells continue to build, we would eventually have a mass, a large mass of cells within which you have the developing primary oocyte. So this is a large mass of cells. What you are seeing here now is referred to as the Graafian follicle. So this is a Graafian follicle, and it is made up of a mass of cells surrounding the developing oocyte. So it is the work of FSH to cause the formation of this Graafian follicle, as well as to cause its enlargement. Now, as this Graafian follicle is formed, it begins to produce another hormone, that is estrogen. So estrogen is produced at first by the Graafian follicle, and that Graafian follicle we said was produced under the influence of FSH. Now, once estrogen is produced, it will have two main functions. One thing estrogen can do is to repair the wall of the uterus. So it will repair the wall of the uterus. Of course, the wall of the uterus must have sloughed off during the end of the last cycle, leading to menstruation at the beginning of this new cycle. So when a woman sees blood, when a woman sees her menses, it is evidence of sloughing off of the wall of her uterus. So after the sloughing off, there's the need for repair, just in case there's pregnancy, so that it can implantation can take place. So we say the estrogen is the one that repairs the wall of the uterus in preparation for pregnancy, just in case it happens. So that's estrogen's first function, to repair the wall of the uterus in anticipation of pregnancy. And then number two function of estrogen is to inhibit FSH. Yes, it has to inhibit FSH. Why? Because FSH has the habit of causing the formation of Graafian follicles. And in a woman, ordinarily, it is supposed to be the case that for every month and in every cycle, only one egg cell is supposed to develop. Only one primary oocyte, is supposed to develop to become an egg, so that eventually there can be pregnancy and all of that. So if FSH is left to just continue to be present in a woman like that, then it means that the primary spermatocytes will be selected, haphazardly, randomly, so that in a month a woman can have up to 10 Graafian follicles developing at the same time. So once one Graafian follicle has started developing and it produces estrogen, this estrogen blocks off FSH so that no other Graafian follicle develops till the end of that menstrual cycle. Then, having seen the functions of estrogen, this guy continues to develop. And as it develops, as it becomes bigger in size, the estrogen it is producing will also be increasing in quantity. Now, somewhere around the middle of the woman's cycle, ordinarily we say a woman has a cycle of about 28 days. So this is like a midpoint, 14 days. This is day 1, so there's no 0 day, and this is day 28. So day 1 of the woman's cycle is usually the first day she sees blood, the first day she menstruates. So if a woman menstruates first, let's say on the 4th of January, 4th of January is the first day she sees menstruation, that day is taken as day 1 of her menstrual cycle. And for a woman whose menses will last for like 3-4 days, let's say 4 days, it means that she will see blood on the 4th, on the 5th, on the 6th, and on the 7th of January. So those are days 1 to 4. Then, during this period, of course, like we said, a follicle would have been secreted or collected, or a follicle has started developing, and that follicle must have released estrogen. So estrogen has blocked off FSH, and estrogen has started the repair of this wall. The wall was degenerated during this period. So estrogen begins the repair of the wall, and as the wall is being repaired, what happens to levels of estrogen? It will keep rising like that. Now there's a level that this estrogen gets up to, such that it will stimulate the anterior pituitary to release its second hormone, which is called luteinizing hormone. The luteinizing hormone is released in a surge, like suddenly. So once that luteinizing hormone is released, which is somewhere around day 14, it may not necessarily be on day 14, but somewhere around day 14, somewhere mid-cycle, the LH is released due to increasing levels of estrogen. Now, once that LH is released, it goes straight to this follicle, and it achieves two important things. Namely, first, the LH causes this follicle to open up so that the secondary oocyte inside it, remember that primary oocyte has undergone some development up to secondary oocyte are rested at metaphase 2. So it is LH that comes to this follicle and opens it so that the secondary oocyte can fall off. Of course, that process is called ovulation. So I'm saying in effect that the hormone that brings about ovulation in a woman is LH, luteinizing hormone. Then, after causing that to happen, so this secondary oocyte is no more, our follicle is now empty. The second function of LH is to convert that empty follicle now into a gland that we call yellow body. Yellow body, in Latin we have corpus luteum. Corpus luteum. So luteum is yellow and corpus is body. So corpus luteum means yellow body. So the corpus luteum is formed from the Graafian follicle after the secondary oocyte has been expelled from it. So expulsion of the secondary oocyte, which is ovulation, is the work of LH. And then also the conversion of that Graafian follicle into a corpus luteum is also the function of LH. So after the corpus luteum has been formed somewhere around mid-cycle, that corpus luteum continues what it was doing before, which is production of estrogen. But not only that, the corpus luteum begins to secrete a second hormone in addition to estrogen, and that hormone is progesterone. So progesterone and estrogen from this point onwards are produced by the corpus luteum. So from what we've said so far, we've seen the work of this guy. It stimulates the anterior pituitary to produce these two. We've seen the work of this one, development of the Graafian follicle. We've seen the work of LH, ovulation and conversion of the Graafian follicle into a corpus luteum. Estrogen, we said first, it causes repair or promotes repair of the uterine wall, and then we said it inhibits FSH. By inhibiting FSH, it ensures that we don't have more than one follicle developing at a particular time. Then, together with progesterone now, from this point onwards, estrogen is able to continue the repair of the uterine wall, thickening of the uterine wall, hoping that there will be fertilization of the secondary oocyte that was released during ovulation. Now, if that secondary oocyte gets fertilized, then of course the wall of the uterus has been prepared for the implantation process. So yes, implantation can take place. But in the event of that secondary oocyte not getting fertilized, then what happens is that this wall would have been prepared in vain. So that by the time we get to day 28 and there's no fertilization, this corpus luteum dies, it ruptures. And once the corpus luteum goes away, once it dies away, it means that levels of estrogen and progesterone will fall. And once their levels fall, that wall that they had been preparing all the while no longer stands. So the wall begins to fall, the wall sheds. And of course, remember we're on day 28. So once there's shedding of the wall, that takes us into a new cycle. Day 1, day 2, day 3, day 4, where we have the wall breaking down. Why is the wall breaking down? Because estrogen and progesterone are no more. And why are they no more? Because the corpus luteum has broken down. And why did it break down? Because fertilization of the secondary oocyte did not take place. So with estrogen gone, one thing that happens is that the wall breaks down, but that's not all. Remember that estrogen blocked off FSH at first so that no more follicles are produced. Now with estrogen gone, FSH is free to act. So what does FSH do now? It will pick another secondary oocyte. We're already in a new cycle now. Sorry, primary oocyte. It picks another primary oocyte and then surrounds it with follicle cells like this. And a new cycle starts. So everything we said here starts all over again. So that's how a woman is able to have, regular cycles where she bleeds, later she ovulates, and so on. Then it's important to add that the release of progesterone from the corpus luteum around this day 14 is usually associated with an increase in body temperature. So for some women, how they know that they have ovulated, how they know that ovulation has taken place, is that increase in body temperature that they experience around mid-cycle. So we are saying that for a woman who observes such increase in body temperature, yes, it can be attributed to the release of progesterone from the corpus luteum. Now, what does an egg look like? Let's end this video with that. For the egg or ovum of a woman, that's the human egg now, we may not be able to describe the exact structure of the human egg, like right here. But the egg of a bird may just suffice, or some other eggs. So this is like a general representation. What I'm drawing here is not necessarily the human egg, but this is a general representation. This is a common diagram we see of an egg. So I'll draw this diagram. Sorry, that's not very straight. Okay, so let's say something like this. And then we have this inside, we have that. Let's just do this. I'm doing that to avoid shading. Yeah, something like this on the average. Then here, maybe I'll just put, okay, something like that. Now, looking at this egg, I'll tell you the basics. Whether you're looking at this egg or some other egg in the future, the basic information is the same. Now, a human egg is surrounded by two major membranes. This innermost membrane here is referred to as the plasma membrane, which is like cell membrane normally that you have surrounding animal cells. And then there's another one here, which is where I have put the line there. You see that zigzag I've put there? That thickness represents the second membrane, which we'll call the vitelline membrane. Then after the vitelline membrane, or outside it, there is a glycoprotein coat that we call the jelly coat. So we'll call it the jelly coat. So it means that coming from the outside of a cell, you have JVP to encounter. JVP stands for jelly coat, then vitelline membrane, then innermost plasma membrane. Those are the coverings that we find outside the egg. And then going into the egg, of course, because it's a cell, we expect that it will have a nucleus. So that's the nucleus of the egg cell. And then the cytoplasm is clearly divided into two in this case. In this case, you see that there's a part of the cytoplasm I'm working on now. That part of the cytoplasm has what we refer to as yolk. Yolk is a protein and lipid material. It has protein, it also has lipid in it. So this part of it, where I'm putting a dot now, that is called yolky cytoplasm. Yolky cytoplasm. And then the other part of the cytoplasm here, where I'm putting another dot, is called the non-yolky cytoplasm. Non-yolky cytoplasm. So there's some part of the cytoplasm where there's yolk, and then there's another part where there's no yolk. But I tell you, if you watch our video on Oogenesis and fertilization, you'd see that there are different kinds of eggs with different kinds of yolk content. Some of them do not have the yolk, the alecytal eggs. Then some have very small amounts of yolk, some have large amounts. And then even when you have different amounts of yolk, we can also talk about the distribution of the yolk. So all of those are terms we'll see in the video under fertilization. But for now, we are assuming here that this egg that is in focus is one that has yolk on one side of the egg, what we will refer to later as a telolecytal egg, and then the rest of the cytoplasm here is clear and devoid of yolk. Now, based on the distribution of the yolk, we say that this egg that is in focus shows polarity. Yes, it shows polarity in that there's the yolky pole and the non-yolky pole. That pole that has yolk, we refer to it as the vegetal pole. So this is the vegetal pole, this one. And then this other pole of the egg is referred to as the animal pole. So the animal pole is the pole without yolk. Then the pole with yolk, the side with yolk, is called the vegetal pole. So those are the two poles. So we say the egg shows polarity, and these are the two poles. It has the vegetal pole and the animal pole. Then these guys you are seeing here are the polar bodies. Remember we said that polar bodies are formed alongside with the egg. So alongside the egg you have the polar bodies, and those are the polar bodies there. So this is a simplified representation of an egg, like a generalized representation. And like I said before, the human egg may not exactly be like this, but it shares many properties, many attributes with what we have on the board here. So if you see the diagram of an egg, I expect that you'll be able to identify those parts. And then all the other things we said about oogenesis, I expect that you'd remember them, and for exam purposes, take note of the terms I used. Watch the video back if necessary, so that on the day of the exam you're able to deliver. That will be all for this video. Thanks for watching. I hope that you enjoyed it, and I also hope that you like, that you share for friends to see. And then if you have not subscribed, please subscribe so that you get notified when I put up new videos. So watch out for the next video, which will be on fertilization and organogenesis. See you in that video.